Thank you very much for that introduction. Notice it's a fool's errand with a question mark. The purpose of the title is to emphasize that some may think that control of PRRS is not possible. So if that's your goal, you're a fool because it is unachievable. I hope to impress upon you that it is achievable and that the scientists who study the problems have something to offer towards that solution. And so one of the things I'll be doing is showing some of the advances in knowledge that some are recent, some are old and being rediscovered that give us hope that we will be able to make progress. I need to disclose that I have PRRS virus patents and I receive consulting income from Boehringer, and that the University of Minnesota manages this in accordance with conflict of interest policies. So PRRS, as you saw yesterday, is tremendously diverse as a virus. There's huge diversity in type 1 viruses that are most common in Europe, huge diversity in type 2 viruses in the rest of the world, and those type 1 and type 2 are remarkably different from each other. So is PRRS just a variety of different flavors of good gelato, or a variety of flavors of this slop that you can get in the United States, or are these two things completely different? And we're committed to the belief that type 1 and type 2 viruses are the same biologically and in most of their properties, so that the research which is done on one informs us about the other. So most of the research is on type 2 viruses, but it's relevant to the type 1 virus problems in Europe. So PRRS is different from other viral infections. Point here is, if that's true, then if you want to understand PRRS, you need to study PRRS virus in pigs. It doesn't do very much to study SIV or FMDV as your model for PRRS virus, because the characteristics of the infection and the interaction with pigs do not help you understand how to control PRRS. Another theme that is becoming very apparent and important to me is the theme of variation. Andrea Ladenig yesterday, I think, impressed upon us how much variation they see in the reproductive disease in PRRS virus in their studies. That is characteristic of all aspects of the interaction of PRRS virus with pigs. The variation is not just in the virus. You'll see there is a lot of variation in the pig in terms of its ability to deal with the virus. And in terms of arriving at tools for controlling PRRS, we need to understand what makes pigs more or less susceptible, as well as what makes the viruses different. Oops, okay. This is a slide that shows the diversity of PRRS viruses taken from field outbreaks in the US. About 5 large producers over 15 years have collected almost 7,000 independent virus sequences. Independent samples from independent outbreaks or the same outbreak at different times. So 7,000 over 15 years gives you an idea of how serious the problem is. There is a lot of variation. So this is at 5%, you've got 15% differences in this group of virus, is the same here. But what's really interesting to me is that you also have outbreaks over a period of years where there's almost no change at all in the virus. This is a completely different concept than you get from all of the scientific papers you can read, where in order to show you how much variation there is, when you take your data set, you get rid of everything that looks similar to something else, and you have a single representative. It misses the point. It says here that there's something else going on in PRRS virus. Are there situations where the virus is so well adapted, or something about its genetic makeup allows it to be maintained for periods of years without changing at all? And this also shows how different the type 1 PRRS viruses are from type 2 viruses, and the amount of variation in type 1 is as much or more as type 2. Second concept in PRRS viruses, scientifically, to study viruses, you clone the virus so that you know exactly what you have, and you determine its sequence so you know exactly what it is. There's only one sequence. It's a clone. By definition, it's all the same. If you look at a PRRS virus sample with high throughput sequencing, which is available now, and so you have tens of thousands of sequences across every position in the genome, shown here, and you can ask, how often do you see one base versus another base? And the frequency of variation is about 0.1%. One base per thousand, you see a difference. So everywhere, almost everywhere in the genome, you see variation in the base that's there. 4.5% of the bases in this example showed no change at all. So there's a lot of variation across the entire genome. A colleague, Dongwan Yu at the University of Illinois, gave us an infectious clone. So again, by definition, this is a single sequence. To get the virus from the infectious clone, you have to put it on cells and grow it. You take the virus from the first passage and you do this high throughput sequencing, it's not a single clone at all. There's genetic variation clear across the genome. It's maintained through passage 2 and passage 3 as well. So this idea, you take PRRS virus in a pig or in a culture dish, it's not a single virus. There is a lot of variation. And if you look in the field, we have 3 different examples of viruses isolated in a blood sample and directly sequenced with no other manipulation at all. And again, you see variation clearly occurring, and you have some positions where there are bases which are, say, 50-50 in distribution. And part of our challenge then is to figure out if these hotspots of variation are significant or just random variation. Oh. Okay. The preceding slide just simply showed that if you looked at some place in the genome, was there variation? But what's more interesting is, are the viruses different? If you have 2 bases that are different, are they in the same genome, or do you just have variation that's random? Haplotypes is a way of asking what are the sets of combinations that are different. So here it's a way of asking, are the individuals variable? And using a method that was recently published for HIV analysis, if we look at this P1 of the cloned virus, what we're looking at here is the frequency of individual haplotypes. So this is one single sequence, which is present at a frequency of 1 in 10,000. And this is a second different sequence. So every position along this axis is a different sequence, and the y-axis is how abundant it is in the population. So looking at ORF5 of PRRS virus or ORF3, we see that this first passage of virus, everything is equally abundant, but there's a lot of variation. If this is 1 in 10,000 here, there's at least 10,000 different haplotypes. In P2, we see that a selection is going on so that there are some sequences that are now more abundant and others that are still present in a single copy. And this distribution is present if you look in, again, 2 different places in the genome. And if you culture the viruses one more time, you see further selection here. 71% of all the isolates are actually a single clone. So what we're looking at is that there is selection pressure when you're growing a virus on the same cells. But as you pass it from dish to dish, you see a change in the distribution of a population. You have this large group of viruses And when you put it into culture, you take a small number of those and start them over. It's the same when you infect pigs. Let's see here. If you look in pigs, you see titers of 10 to the fourth or 10 to the virus per mil. Pigs have a couple liters of blood. There's billions and billions of viruses there. And it's a large population of individuals that vary just like you vary in this room. When a new pig gets infected, it's a very small number of viruses that goes. So there's a huge bottleneck where you're collecting just a subpopulation that may not be representative. So here in this example, 45% of the sequences were this single virus, the next most abundant 13%, and so on. And then you have this large number of very rare sequences. And you see this regardless of where you look in the genome. So this is a different idea when, so when you take, let's go back here. If you take 10 or 100 of the viruses out of this whole population, what's that going to look like in the next pig? That experiment was done about 14 years ago in Jeff Zimmerman and Kunjin Yoon's lab at Iowa State, where pig in line A, passage 1, they started with a cloned virus, VR2332, which in their mind was all identical. And they took out, after 60 days of infection, they harvested viruses out of the tonsil, sequenced 15 isolates, and looked at what they were. Of those 15, 6 were the original sequence, 7 were different. There was one sequence that was unique, and then a number. So every new sequence they saw, every haplotype, they gave a new designation. They repeated this experiment multiple times. So this mixture of viruses was then used to infect in the second passage. In the second passage, what they started with was now gone, and they had a dominant sequence here that was from here. They took this sample, put it into a third pig, and a fourth pig, and a fifth pig, and by the seventh passage, they had this distribution right shown here. So you can see that every time they passaged the virus through pigs, what came out was not identical to what came in. And it looks random because they repeated this in a second line of pigs, the B, and here you see the results vary from A. And in the second passage, it was a completely different set of clones, and in the third passage, it was again, and they did this a third time, the C line of pigs. Here's what came out after the first passage, the second passage, and again, the third passage. The seventh passage. So the point here is that the sequences are constantly changing genetically. They also look to ask if there were antigenic changes, because it's just assumed that when viruses change in pigs, that it's immunological selection, and it's going to lead to escape mutants. It's just ignored that no one in 25 years has ever actually held in their hand a virus which has escaped the immunity of the previous pig and now is resistant to it. And when they looked at antigenic changes after these 70 passages, which 7 passages taking 367 days of virus growth in pigs, they looked for antigenic change by testing their reactivity to monoclonal antibodies to nucleocapsid and E protein, and found no changes at all in the ability of the serum to neutralize the viruses. So again, we have evolution going on here, a selection process, but no evidence that it's immunological. And this slide shows that you see the same kind of thing in commercial herds. data provided by Dale Poulsen. You heard his name already. He's taken in vain sometimes by people who like shortcuts, because Dale is nothing if not exhaustively thorough. So here, Dale's plan was to do PCR in a large system weekly for 7 years, 371 weeks. Every PCR positive got submitted for sequencing. And in the first week, the sequence of a sequence of a virus was found. I don't know if everyone can see this. And this is how different a sequence is from what was started with. So it was itself. The next week and succeeding weeks, you can see that the sequences of this virus, this was never found again. What you had here was a cluster of variable sequences that were 0.8 to 2.2% different. And over a period of several years, they didn't change until finally they started gradually increasing in diversity. This is one pattern. In this same system, a second virus appeared. It was introduced into the herd 40 weeks later. And you can see here that over time, the same sequence was isolated for more than a year. The identical sequence, the same haplotype. In addition, you could see other variants. And then there was this jump in which it continued to increase. So here we have 3 replicates in a commercial herd, 3 different patterns. But what's really clear here is that when you infect pigs, it's a small number that becomes a huge number, and when there's transmission events, it's a small number again that's retransmitted to another pig. So you have this huge mixture of viruses that are picked perhaps at random, perhaps based on ability to be transmitted, and that's what's driving the variation we see as far as we can tell. So we are learning new things about the PRRS virus that help us in our understanding to try and control it. It's a population of mutants, whether you're looking in pigs or in cell culture. Genetic variation is distributed across the entire genome, and that's confirmed when you do genome sequencing and compare your sequences. Evidence for immune selection is lacking at this time. And so if a virus is evolving, and it for sure is, what are the drivers of evolution? Is it ability to be transmitted? Is it just a random process? Okay. I'd like now to move away from the virus to the pig, talk about innate immunity for a little bit, and then talk about adaptive immunity. And adaptive immunity is where memory comes from, and memory is the basis by which vaccines work. So we want to pay attention to it and to understand it. So it's obvious that there's a lot of variation in how pigs respond to PRRS virus, and it's well known now that there's an age-dependent immunity shown here, where if you infect weaned pigs with a virulent virus, it goes to a high titer and lasts for weeks. Same virus put into sows grows much less well for a much shorter period of time. Sows are more resistant to infection than weaned pigs. Same response with vaccines, which are attenuated viruses. This is vaccine growing in weaned pigs versus sows. So what's the basis for this? PRRS virus grows in macrophages, and it turns out that the viruses grow. I may have skipped a slide. PRRS virus clearly grows better in young macrophages than older macrophages, whether you're measuring it by how much virus is in the cells. So in high throughput sequencing, the number of reads is proportional to the number to the abundance of virus. Young, and you see how much variation there is versus adult. If you look at the percent of infected macrophages. Young macrophages are more susceptible to being infected than adult macrophages. And if you look at how virus grows in cell culture, it grows better in young pigs than in old pigs. So what's going on here? The most obvious first question would be, well, the receptor that enables virus to get into the cells might be more abundant on young macrophages, but it's not. CD163, which is all you need to infect macrophages in pigs, abundance is the same in young and old. To look more deeply, we went to a high-throughput genetic method of sequencing the RNA, which is the expressed genes of macrophages, and analyze them by something called principal components, which basically asks if there are global differences in your comparison groups. And if you compare the expression patterns in young versus adult macrophages, they're different. And if you look at infected cells versus uninfected cells versus infected, you see that they're different. So then the next step is to ask, what are these differences due to? And here we use volcano plots. When I was introduced to volcano plots, oh, it's a neat word. It sounds really neat, and you can imagine stuff spewing out, but it doesn't really explain what you're looking at. So we're comparing the expression levels of genes in uninfected adults compared to infected, and so on the right side is infected adults, where whatever this gene is, there's a lot of it in infected, and it's somewhere down really low in uninfected. The point here is that in infection, you see a lot of genes turned on, including this one up here. The top 10 list is shown. Interferon beta is the most expressed gene you see when you infect macrophages. To me, this is really, really neat. We're looking at the naturally permissive cell and PRRS virus. There is 10 to 15 years of research where my colleagues have picked out different genes from PRRS virus and put them into artificial model systems and looked to see if they suppressed interferon responses in responding cells. And they always block expression. So the idea now is commonly held that PRRS virus suppresses the innate immune response. And we see when you actually look at what's happening in pig cells, the most elevated gene is interferon beta. Then another interferon, alpha omega, is the next highest, interferon alpha 4 the next. Oligoadenylate synthase, a interferon response gene, is next, and so on. There's this huge interferon response. How does this compare then in adults to young pigs? In young pigs, we see the same type of response. Interferon beta is the highest expressed gene of all. Interferon alpha omega, the same genes pop up again. The difference here is that this level of expression is 20 times lower than it is in adults. So it looks to me like the adult macrophages just mount a much stronger innate immune response. Oh. And this is called a pathway analysis. It's real easy when you just look at global gene expression to find something that's really high. To determine if it's important, you take into account the fact that genes act in pathways that make things happen. So if the pathway is turned on, then you have strong evidence that the process is actually going on. Here, interferon beta is the most turned on gene following PRRS virus infection, and the viral infections are known to have turn on a cascade of genes that send signals all the way down and turn on interferon and their response genes. And all of these colored genes here are those that are turned on in the infected macrophage. So we have really good evidence that, in fact, this is a major innate mechanism of resistance to PRRS virus infection. I think that this explains then some of the experiences we and other people have had trying to culture PRRS virus out of serum samples in vitro to isolate the virus and study it. A lot of efforts to grow viruses fail. This is data published from our lab where we had 50 field virus isolates over 14 days culturing them on the the simian cell line MA104. We only got 2 out of 50 to grow. When we put these on macrophages, we did much better, whether we're measuring by fluorescence, which indicates virus proteins in the cell, or cytopathic effect, or viral RNA copies at different times. We see better results, but still 30% of the isolates never grew at all. Why? Well, you've got this interferon beta response that actually works. You have variation in virus, but you have variation in pigs, where some pigs control infection better than others. So we are learning about innate immunity. Pattern recognition receptors sense PRRS virus and they mount an oftentimes effective antiviral response. This response is better in adult macrophages. If you're vaccinating piglets, it's likely that the vaccine may be more effective than in adults where you have this innate immune response in the sows that's more robust than in the piglets. PRRS immune countermeasures do not block porcine immune responses completely, not effectively. Model systems for virus cell pathogenesis and immunity, in my opinion, do not translate to studying PRRS virus in pigs. So there is an innate immune response to PRRS. We are not on a fool's errand. Okay. So in the closing minutes, I'd like to look at the adaptive immune response. I'm going to concentrate on neutralizing antibodies, not because we're convinced that they're the key to controlling viral infection. We know that you can control viral infections without any neutralizing antibodies at all, but they play a key role in controlling viral infection in many species against many viruses. So it's a reasonable assumption that if you can get better neutralizing antibody responses in the pig, you'll be able to improve protection. This is a standard illustration of the lack of neutralizing antibodies in PRRS virus responses. Studies done in young pigs where you take serum from pigs infected with different viruses and you test neutralizing activity against the same virus or all the others. So here we're looking at homologous neutralizing activity where we see variation. We see some. These titers are not dramatic, but they're real, and we see none. And we see numbers in between. Heterologous cross-protection, we see very little neutralizing activity. And heterologous immunity is how vaccines work. Vaccines are attenuated viruses taken from a point in time in the field, and years later they go on the market and they're used for years afterwards. That original virus sequence does not exist anymore, so vaccines only work by able to provide cross-protection or heterologous protection. Okay. So that was an example, one of many taken from growing pigs, where it's hard to see neutralizing antibodies. A few years ago, we went and got blood samples from cull sows in herds that had suffered PRRS virus outbreaks. And to our surprise, we had high titers of neutralizing antibodies. This is 20 pigs from herd 1 looking at average and variation, 14 pigs from herd 2 and herd 3. Every herd we look at, we see a large number of pigs with substantial amounts of neutralizing activity. 50% titers here, 1 to 20 to 1 to 300, where in young pigs you're looking at 0 to 1 to 8 typically. Okay. We're looking at cross protection here. Wild viruses cause the outbreaks here. The test virus and neutralization assays is a lab strain VR2332, sequence difference of about 15%. Okay. These sera show a wide range of neutralizing activity. Collaborator Eric Nelson at South Dakota State ran these assays in his neutralization assay system. And again, we see substantial neutralizing activity against a variety of type 2 viruses. Most interesting was neutralizing activity against type 1 viruses, which the herd had never seen. Obviously, they're higher. The reasons for this might be real, which would be interesting. It might be a subtle technical explanation of how the assay is done, because the type 1 viruses grow more slowly in cell culture than type 2. This neutralizing activity is functionally relevant in vivo. It provides passive cross protection against virulent virus infection. Here we're looking at reduction in viremia of a type 1 PRRS virus infection from passively administered pigs, and a much more significant reduction in viremia from a type 2 virus infection. This cross protection is easy to see in studies in the field. There are, well, Boehringer Ingelheim has done lots of challenge studies. Every time there's a new virus outbreak, they do their best to acquire a virus from those outbreaks and do an experimental challenge to see if their vaccines still protect. And in the examples that they have presented in various venues and meetings, always shows substantial protection. What's more interesting to me is that scientists who question whether cross protection and heterologous protection are real, everyone who has ever at some point done a challenge study has also seen that cross protection works. Here we're looking at protection of the pigs immunized with the MLV vaccine against challenge with high-path PRRS virus in China, the most virulent virus that you can have. In many examples from a variety of labs, it kills 100% of tested pigs. Here it killed two-thirds of the pigs. Survival after 21 days was 4 out of 12. Vaccinated animals, 100% survived. They still had disease. They had fevers that are significant, but they're much reduced from non-vaccinated. And this simply shows the survival. These really virulent viruses kill pigs as little as 6 or 7 days after infection. Okay. We go back to, again, neutralizing activity. We're interested in the targets of neutralizing antibodies. If nothing else, if you identify a target, it becomes a candidate immunogen that by itself could provide protection against PRRS. The There are dozens and dozens of papers in the literature, some of which I have contributed to, that suggest that the major envelope glycoproteins, GP5 and M, are the targets of neutralizing antibodies. A student in my lab, Juan Lee, coated beads with recombinant proteins for GP5M, the major heterodimer on the surface of the virus, or GP5 or M, showed that these beads would bind GP5 antibodies, and then asked if those antibodies would neutralize. So those antibodies bind virions, shown here, so they work, but they do not neutralize activity. So this was one of 5 different ways that Juan Lee showed that GP5 was not the target of neutralizing antibodies, that vaccines that are in the field based on, in some countries, that are based on viruses that express GP5M are not going to work. And the virologist, particularly Fernando Osorio at University of Nebraska, has good evidence now that the true targets, the receptors on the virus that are probably the targets of neutralizing antibodies, are minor envelope glycoproteins GP2, 3, and 4. And Simon Graham showed a picture of that yesterday, where the artist's concept of how those proteins bind CD163 on the virus. Okay. So regardless of what the mechanism of protection is, where neutralizing antibodies fit, and what the target is on the virus, we know for sure that immunity to PRRS virus is complete, and you can sterilize animals, get rid of all the virus. We know this from herd closure. I assume all of you have heard about this now multiple times. Again, you expose all of the animals in a barn to virus, you close it, you bring in no naive animals that might be able to maintain pockets, and after several hundred days, you open up the barn and there's simply no virus at all. We heard the example of the pipestone systems, where their goal is to wean negative pigs forever. When they get a break, they do herd closure to go negative again. This really works. So it tells us scientifically that pigs have the ability to get rid of the virus. It takes a really long time. We have to figure out how to make it work faster, and we have to look at the individual pigs to figure out issues of variation. Here we're looking at plasma cells. So antibodies are made from B cells. Plasma cells are actively secreting antibodies, and in following a PRRS virus infection, we see these cells peak at the end of acute infection and then decline rapidly. So these are the cells producing the antibodies that you see in your IDEX ELISA nucleocapsid. But whatever viral protein you're measuring, you see that a broad immune response is generated. Okay. Memory cell responses are crucial for vaccine effectiveness, and here we see memory cells persist for long periods of time. That's the concept, and it works. So there is a strong immune response. It's not interfered with. PRRS virus is not blocking the ability to make an immune response. One of the characteristic features that is in common in textbooks is that if you reinfect, you get a huge anamnestic response. And one of the characteristics of PRRS virus is this does not happen. So here we're looking at antibody responses to different viral proteins following infection. And then at the arrow, animals were challenged. And you see in no case at all is there an anamnestic response. And we're looking at the colored lines are averages from 10 pigs, each infected with a different virus. You also see that nucleocapsid shows this characteristic peak and decline, which oftentimes results in animals going seronegative. So the issue here is what's going on? Why is there not an anamnestic response? Is it because the pigs cannot respond, thus questioning the ability of vaccines to protect, or is it something different? And what we show here is that this memory response is fully armed and ready to act. We're looking at antibody secreting cells in a recall assay from pigs infected one time with the virus, and so these are the responses you see in different lymphoid tissues. If you challenge in vivo, like we just saw, you see no change in response. But if you take the cells out of the pig and re-stimulate in vitro, these cells are fully capable of mounting a large response. So there has to be an explanation for why this happens. One explanation is that the immune resistance to PRRS virus is so good that the virus cannot grow, and so there's no antigen to re-stimulate these cells. And I think this is the last data slide. And it comes back to the issue of variation in the pigs. These are pigs. We're looking at groups of 10 pigs. Each group got infected with a different virus, and we're looking at lung scores at necropsy from individual pigs. And you see, it's obvious some viruses are more virulent than others, but in every single group, even here where one pig died, another pig was completely protected, no lung lesions at all. So what's the basis for these high levels of variation in the pigs? Is this the pig that we would like to select in understanding how it resists immunity? And do our genetic selection or our nutritional benefits or whatever on this genotype compared to these others which are clearly more susceptible? And our focus is on the neutralizing antibodies and how they're made, and is there genetic variation at the immunoglobulin loci that are contributing to resistance to PRRS. So overall conclusions, it's clear to me our concepts of what PRRS virus is needs to change. It's a large population of viruses undergoing genetic bottlenecks every time you have transmission from one pig to another. Innate immunity is driven by interferon beta and stronger in adult pigs. Broadly neutralizing antibodies, memory, and herd closure together show that immune protection is real. Not absolute, but it's real. Variation in the host pig, including genetic and other factors, increases the challenges in finding solutions. But for all of these reasons, control of PRRS is not a fool's errand. Thank you.
Thank you, Mike. We have time for one short question. We will have more time at the end before we go for our coffee break. And maybe people need to think about this for a while before asking a question. I have one if there's not one from the floor. Mike, how all of this knowledge, which is quite overwhelming, how do you think it's driving the decisions on how to approach PRRS? Do you think in the United States in particular, people are moving more towards elimination or control?
Well, it was about 8 years ago when Scott D. led the push for a national PRRS elimination program. And then about the time that the area regional control projects were started, I'd say 5 years ago, the focus was on control, not on elimination, because elimination was viewed as too difficult. There were conditions, so there are examples where PRRS virus was eliminated from a region, a county in Minnesota, a system in South Dakota, and the next year new viruses were introduced, and so it turned out this elimination was not permanent. The control is a much more, it's a goal that is perceived as being more attainable, and the tools are there. The vaccine provides a level of control. Disease outbreaks are not as severe, but the strongest argument for using vaccines is economic arguments where people have done cost-benefit analyses of doing nothing, managing outbreaks with vaccination, or managing outbreaks with live virus inoculation. And there was a veterinarian for a large system made a presentation a couple years ago showing the cost benefit of vaccine over live virus elimination or inoculation in their system. And this was on a Saturday morning, and you could literally feel people running out of the room to call back to order vaccine. The economics really drives decision making in the US, and that's one of my feelings about PRRS in Europe is that producers do not really understand the cost of PRRS. It's in part because these economic analyses are not commonly done.
Okay, with that very good comment for us, we will move to the coffee break. I believe we have planned until 10:25.